Hello friends, today's topic is various models of plasma membrane. In eukaryotic cells, the plasma membrane surrounds a cytoplasm filled with ribosomes and organelles. Organelles structures that are themselves encased in membranes. Some organelles like nuclei, mitochondria, chloroplasts are even surrounded by double membranes. All cellular membranes are composed of two layers of phospholipids embedded with proteins. All are selectively permeable, that is semipermeable, allowing only certain substances to cross the membrane. The unique functions of cellular membranes are due to their different phospholipid and protein compositions. Decades of research have revealed these functions. The four historical models of plasma membranes are lipid and lipid bilayer model in the year 1925 to 1926. Second one is Danielli model, that is in the year of 1935. Then third one is unit membrane model, that is protein lipid bilayer protein in the year 1953. Then the last model is fluid mosaic model, that is in the year 1972. So lipid and lipid bilayer model. In 1902, Overton observed that substances soluble in lipid could easily selectively pass through the membranes. On this basis, he stated that plasma membrane is composed of a thin layer of lipid. Gorter and Grendel in 1925 were the first to suggest a possible structure of the cell membrane. On the basis of studies, cell membranes of erythrocytes, they showed that the lipid extracted from the red cell ghost spread in area twice that of a simple molecular film. So it was thought that the membrane consists of double layers of lipid molecules. The polar hydrophilic groups of the molecules being situated on the outside hydrophobic ends standing at right angles to the surface are oriented inwardly. These models of Gorter and Grendel could not explain the proper structure of plasma membrane, but they put the foundation of future models of membrane structure. Danielli model, sandwich models. James Danielli and Hugh Davison in 1935 have suggested bimolecular leaflet model of cell membrane. So, was the first attempt to describe membrane structure in terms of molecules and to relate the structure to biological and chemical properties. According to biomolecular model of Danielle and Davison, plasma membrane consists of two layers of phospholipid molecules. A biomolecular leaflet in which phospholipid molecules are arranged in such a way that hydrophilic heads of the phospholipid molecules face outside and hydrophobic nonpolar lipid chains are associated in the inner region of leaflet. The hypothesis also suggested that the polar ends of lipid molecules are associated with monomolecular layer of globular proteins The plasma membrane would this consist of a double layer of phospholipid molecules sandwiched that is between essentially continuous layers of protein. The model was also described as a lipoprotein sandwich, as the lipid layer was sandwiched between two protein layers. The Davison-Danielli model predominated until Singer and Nicholson advanced the fluid mosaic model in 1972. The fluid mosaic model expanded on the Davison-Danielli model by including transmembrane proteins and eliminated the previously proposed flanking protein layers that were not well supported by experimental evidence. Unit membrane model. In late 1950s, Robertson summarized a large number of ultrastructural data obtained by him and some other workers and concluded that the plasma membrane and the membranes of the cell organelles were similar in structure. Although the similarity is not resolved by light microscopy, it is clearly seen in electron micrographs. This conclusion led Robertson in 1953 to propose in unit membrane hypothesis, according to which all biological membranes show generalized unit membrane construction. The unit membrane model visualizes cell membrane as a trilamellar and indicates structure consisting of two dark osmophilic layers separated by a light osmophilic layer. The physical appearance of this trilamellar model has led to the term unit membrane. The unit membrane concept implies a trilamellar appearance with a bimolecular lipid layer between two protein layers. Each dense osmophilic band is made up of protein and the polar groups of phospholipids and thus is 25 angstrom thick. The clear osmophilic zone 35 angstrom in thickness is a bimolecular layer of lipids within the polar groups. In other words, the unit membrane is 75 angstrom thick with a 35 angstrom thick phospholipid layer between two 20 angstrom thick protein layers. The plasma membrane surrounding the cell is thicker at the free surfaces of the cell than where it is in contact with other cells. In unit membrane model, the protein layers are asymmetrical. On the outer surface, it is mucoprotein, while on the inner surface, it is non-mucoid protein.